Hey guys, welcome back. I hope everyone is doing good. In today's video, we are going to discuss Euglena's morphology. So let's begin. Okay guys, so like always, we should start with the systematic position. Euglena belongs to phylum Protozoa, subphylum Sarcomastigophora, superclass Mastigophora and class Phytomastigophoria. Now Euglena belongs to phylum Protozoa. You know protozoans have unicellular eukaryotic bodies. Subphylum Sarcomastigophora includes individuals which have either a flagellum or pseudopodia, which they are going to use to move from one place to another. Members of superclass Mastigophora have a flagellum, have a whip-like flagellum. As you can see in this diagrammatic representation, they have a whip-like flagellum, which is going to be their locomotory organelle. So Euglena belongs to superclass Mastigophora, and the characteristic feature of members of superclass Mastigophora is this whip-like flagellum. Its class is Phytomastigophoria. Now you should know that the superclass Mastigophora can be divided into two classes. First is your Phytomastigophoria. The second one is Zoomastigophoria. Phytomastigophorians have a chloroplast inside their cells. Here the term phyto stands for chloroplast. So because of this chloroplast, they are able to synthesize their own food by the process of photosynthesis. The zoomastigophorians do not have this chloroplast, and thus they are heterotrophic organisms. They cannot synthesize their own food. On the other hand, phytomastigophorians like Euglena, because they have a chloroplast, they can synthesize their own food, so they are autotrophic. The order is Euglenidae, genus is Euglena, and the species we are going to use as a model organism in this particular video is Euglena viridis. And this picture in fact is of Euglena viridis. So here I have written it's a unicellular flagellated protozoan. It contains a chloroplast and is able to synthesize its own food by photosynthesis. Let's talk about its habit and habitat. So Euglena is a freshwater flagellate. That means that it lives in fresh water. It freely swims in fresh water and it shows solitary existence. That means that it does not live in colonies. It's a solitary organism. Now it's obvious that it's going to be found in pools and ditches which have a good amount of vegetation, because that's going to support its growth and provide it nutrients. Now sometimes it can become so much abundant in ponds that it results in a green scum which covers the surface of water. Now this green scum is not exactly the scum. You know that the scum is made up of algae, right? But it looks like green scum because of the abundance on the surface of the lake. Let's talk about its shape and size. You can see from the picture in reference that it has a very elongated body which is spindle shaped. This anterior end is blunt and this posterior end is pointed. You can also see this long whip-like flagellum originating and coming out from this anterior end. Now let's talk about a very important feature of Euglena, and that is its pellicle. Now pellicle covers the entire surface. Okay, the pellicle is like a thin, rigid, flexible membrane which is present underneath the plasma membrane. You cannot compare pellicle with cell wall by any means. Why? Because you know cell wall is present outside the plasma membrane, but pellicle is present underneath the plasma membrane. Secondly, the pellicle is made up of proteins. You know that cell wall is made up of carbohydrates. For example, if you talk about cell wall of plants, it is made up of cellulose. If you talk about cell wall of fungi, it is made up of chitin and so on. So overall, the nature of cell wall is polysaccharide, or you can say it's carbohydrate in nature. But pellicle is made up of proteins, that is, it is proteinaceous and it is present underneath the plasma membrane. So the outermost limiting layer of Euglena is going to be the plasma membrane, underneath which you are going to find the pellicle. Now it's very thin and rigid, but at the same time it's quite flexible, and because of this actually Euglena is allowed to do movement. It is because of this flexible pellicle that the Euglena is able to show body movement, and as we are going to study in another separate video lecture, Euglena shows a special type of movement called euglenoid movement, which is because of this flexible pellicle. Pellicle is made up of helical strips. These helical strips fuse with the cell body at both ends. So if this my highlighter is going to go over one of the strips, right? So this is one helical strip. So you can see how it is fusing at both ends. Now each of these strips bears a groove along one edge and a ridge along the other edge. And when these strips are placed together, the ridge of one strip fits into the groove of adjacent strip. In order to understand this, you might as well think of a soap box. This is your box and this is your cover. The strips also fit in one another to form this entire pellicle. Now these strips are supported by the microtubules. You know microtubules are cytoskeletal elements. They are present underneath, and these strips are in fact supported and held in place with the help of these microtubules. Now when you look at the pellicle under electron microscope, these spiral strips appear as striations and they are called a special term called myonemes. So this is a very important term, myonemes, which is a feature of pellicle. It basically refers to these spiral strips which are covering the entire cell body of Euglena. Now let's talk about its cytoplasm. The cytoplasm of Euglena is differentiated into two zones. You have the peripheral or the outer ectoplasm and the inner endoplasm. The ectoplasm contains microtubules, which we were talking about, that support the pellicle strips, and it also contains muciferous bodies. These muciferous bodies secrete mucin, and this does the work of lubrication. The endoplasm is obviously the fluid central zone, and it's the one which has all the organelles. So nucleus, endoplasmic reticulum, Golgi apparatus, all of these are suspended in this endoplasm. So the cytoplasm is differentiated into the peripheral ectoplasm and the central endoplasm. Now let's talk about the reservoir, which is a flask-shaped cavity located at the anterior end of the body. Now reservoir, if you talk about reservoir, you also talk about several other features. So over here you can see this flask-shaped cavity. This flask-shaped cavity is the reservoir, but this tunnel-like structure, that's called the cytopharynx or the cell gullet, and this opening is called the cytostome or the cell mouth. This reservoir is also known as the flagellar sac because it holds the flagella. Now looking at these two terms, the cytopharynx and the cytostome, it might appear that it has got to do something with ingestion of food. However, Euglena does not ingest solid food. It does not show holozoic mode of nutrition. The cytostome and the cytopharynx are actually used as a canal for escape of fluid from the reservoir. Now to understand why they are used as a canal for escape of fluid, you'll have to also look up to this structure. This particular structure you can see here is called the contractile apparatus. Now what does the contractile apparatus do? It takes excess fluid from the Euglena cytoplasm and it dumps the excess fluid into the reservoir. From the reservoir it goes into the cytopharynx, and then from the cytostome it is thrown outside the Euglena's body. Now another point to note here is that the reservoir is lined by a plasma membrane only. Here the pellicle is going to be absent. Now let's talk about the flagellum. So Euglena uses a whip-like flagellum to move from one place to another. Now there is a pair of flagella present, right? There is this one long whip-like flagellum which appears outside. It emerges outside the body, and this smaller one which remains confined within the reservoir. These flagella originate from two tiny granules which are called blepharoplasts or kinetosomes. So you can see these granular structures which are embedded in the reservoir, right? They are embedded in the base of the reservoir. These are called the blepharoplasts, and from here the flagella originate. Smaller flagellum, as we just discussed, remains confined within the reservoir, whereas the larger one emerges out through cytostome. Now a flagellum has an outer contractile protoplasmic sheath and an inner elastic axial filament which is called axoneme. Now if you look at the TS, transverse section of a flagellum, this is going to be the structure you're going to see. So you can see these 9 doublets, 1, 2, 3, 4, 5, 6, 7, 8 and 9. So these are called the peripheral fibers, and each peripheral fiber is made up of 2 subfibers. So these are the peripheral fibers. Each peripheral fiber is made up of 2 subfibers, and from one of the subfiber in each pair there are arms originating. So you can see this is a arm, and these arms will face in one direction. So you can see that all of these arms are facing in one particular direction. In the center you have two central fibers, but these are singlets, they are not present in pairs. Around these central fibers you have an inner sheath, and around these peripheral fibers you have the outer sheath. The outer sheath is continuous with the plasma membrane. Now Manton suggested that there are hair-like contractile fibers which are called mastigonemes, which originate or arise from two of the 9 peripheral fibers. That means that from any two of these 9 peripheral fibers, there are going to be these hair-like contractile fibers arising, and he called them mastigonemes. Such a flagellum is called as stichonomatic flagellum, the one which has hair-like contractile fibers called mastigonemes originating from peripheral fibers. That flagellum is called stichonomatic. Now let's talk about its photoreceptor apparatus. Now because Euglena is sensitive to light, it uses light in fact to synthesize its own food. It's going to be an apparatus which it will utilize to orient itself towards the direction of life. And in fact, that particular apparatus is called the photoreceptor apparatus. Now the photoreceptor apparatus has two components. It has a stigma or the red eye spot and a paraflagellar body. The stigma or the red eye spot, as you can see over here, this structure is a red colored cup-shaped structure which is made up of a plate of lipid droplets. Inside each lipid droplet is a carotenoid pigment, and the red color is actually the result of the carotenoid pigment. It is because of this carotenoid pigment that this stigma appears red in color. Now these lipid droplets serve as a lens and they help to focus the light. Now the second component is the paraflagellar body. As the name suggests, para means lateral and flagellar here is for flagellum. So you can see over here, this is your paraflagellar body, and the paraflagellar body is actually, it can either lie on one root or it can lie on the junction of two roots. In this picture you can see that the photoreceptor is lying at the junction of the two roots, but it can also lie towards the lateral side of the longer flagellum. So this is your longer flagellum. So this can also be your position of the paraflagellar body, that is on the lateral side of the longer flagellum. That is why it's called as the paraflagellar body. So let's talk about the contractile vacuole. I already discussed it while I was addressing the reservoir, but let's talk about it in detail, a bit detail now. So its function is osmoregulation. We already saw that it's going to regulate the levels of water in the endoplasm. So that's its function. Now it lies near the reservoir on one side. We already saw its location. The contractile apparatus is made up of a large central vacuole which is surrounded by several minute accessory contractile vacuoles. And it turns out that these accessory vacuoles are the ones which take up the excess fluid from the cytoplasm, and then they basically fuse together to form this central vacuole, and the central vacuole then ultimately flushes all of its content inside the reservoir from which it goes to outside. Now we come to the nucleus. It's very simple. Euglena has a single, round and a large nucleus. It contains a central body which is called as endosome or nucleolus, and the nuclear membrane is a double membrane which is perforated by pores, and inside the nucleoplasm you have the chromosomes present which are thread-like. Now we come to one of the most important features of Euglena, and that is your chromatophore, also known as chloroplast. And the chloroplast of Euglena are arranged in a star-like manner, in a stellar manner. So you can see this is a chloroplast, this is another chloroplast, and all of these are chloroplasts. So they are arranged, as you can see, in a star-like manner, a star-like grouping, and that looks very beautiful. The chloroplast contain the chlorophyll A, B along with beta carotene, which are these photosynthetic pigments also found in the higher plants. Now at the center of each chloroplast there is going to be a pyrenoid body. This pyrenoid body is going to be the center for synthesis of paramylum. Now paramylum is a starch-like substance. It's the reserve food material of Euglena, and it's similar to starch. It is also a polysaccharide, but it's not identical to starch. Okay, it's similar to starch, it's a polysaccharide, but as we are going to see, it has got a different linkage, so that's why it is different. But you should know that this is the reserve food material of Euglena, and it is synthesized in pyrenoid, which is present at the center of chloroplast. Now if I see, this is my chloroplast, and the matrix inside is called the stroma, and in the stroma you will find thylakoids or lamellae, which are actually the ones bearing the chlorophyll. Apart from this, they also have ribosomes and fat globules. And a chloroplast is bound by a triple membrane envelope. This is unlike plant chloroplasts, which you know are bound by a double membrane envelope. Now, as we were discussing paramylon bodies just now, these are found scattered throughout the cytoplasm. So you can see these spiral things, they are in fact your paramylon bodies. They are the ones which are going to store paramylon, which is the reserve food material of the Euglena, and it's going to be the product of photosynthesis. We saw how it was being synthesized in the pyrenoid present in chloroplast. Now it's a polysaccharide, as I mentioned, but it is different from starch because it has got beta 1 to 3 glucan linkage. You know starch has alpha 1 to 4 linkage at the linear chain and alpha 1 to 6 linkage at the branch points. So it's got a different linkage than the starch, so it's not identical to starch. However, it is starch like. It also gives no color with iodine. Starch gives blue color with iodine, but paramylon gives no color with iodine. Now, apart from all of these specialized structures that we just studied, there are other cytoplasmic contents as well. You know, Golgi apparatus is going to be there, endoplasmic reticulum, mitochondria, ribosomes. You're going to have free ribosomes which are going to be scattered in the endoplasm. You're also going to have ribosomes which are associated with the ER, and you're also going to have ribosomes in the chloroplasts. So that's it for today, guys. Thank you for watching. We will meet in our next video.